Thanks for joining us for the latest MCLS talk. Today we have Dr. Eric Klauchik here with us to talk about statistical analysis of network data.
Okay, thank you, Pete. Thank you for the invitation. Thank you, Julie, for helping set this up. I have bounced off and on with MITRE people at conferences periodically over the years, but this is the first time I have the pleasure of being here, so I appreciate the invitation. Let me tell you a little bit about my background just to set context. So my doctoral work is in statistics. Prior to that, I had mathematics. I did about 10 years in signal and image processing, so I've always had a foot in engineering. And for the last 15 years, I've been working in network analysis, which if you have familiarity with it, then you know that that's involved people in statistical physics and mathematics, statistics, and whatnot. So it's been a great deal of fun. I enjoy spending my career sort of mixing with lots of different folks. I'm assuming that the group I'm speaking to, both here in the room and online, are a very mixed group. So what I have here is a sort of an overview of a number of cutting-edge topics in network analysis from a statistics perspective. My goal is to try and give you a little bit of a sense as to things that you might figure we all know how to do with statistics in general, but where when it hits networks, it actually is still stuff we're working on. And so subtitle there is visiting the foundations, because network analysis is already about a 20-year-old field in its modern form, and yet we're still really actually laying a lot of the foundations in statistics. Okay. All right, so I'm going to give you a little bit of background here, particularly for those who maybe haven't spent a bunch of time in complex networks. And then I'm going to go through three what I call vignettes, right? So these are just to be quick, small, focused problems where I'll give you a little bit of sense as to what the challenge is, a little bit of sense of what we've done on it, and a sense of sort of what are still some of the open areas that we're working on. Okay, so complex networks. I had a colleague ask me once, so network analysis, is that mainly just producing these big fancy pictures, or is there anything more to it? So I guess we have that coming, because certainly anyone who talks on networks has some sort of fancy pictures they show. These are visualizations of a small gene regulatory network. This is an attempt to visualize science and the collaborative nature of it. And that's actually an attempt to visualize the router level internet, aka around 10 years ago or so. So this has been a very small field of study until about 20 years ago when it really exploded. It's had an epidemic-like growth. And arguably, you know, we can talk over a beer or a glass of wine or something as to why, but I would say systems level perspective in science is one of the major reasons that complex networks has really taken the world by storm. When you think about systems, you think about interacting pieces, a graph is a way to represent them. Boom, you're off into networks, right? If you didn't have any data to go with the system level perspective, then it's all conceptual. So the movement towards a flood of high throughput data in all kinds of different fields, from biology to, I was just telling Pete today, we're working now on high throughput chemistry at BU. This ability to measure simultaneously the elements across the system, sometimes continuously in time, that's also a major contributor. Right? So a lot has happened in 20 years. Every once in a while I try and update my numbers. These are maybe 6 years old, but Google Scholar says there's over 500,000 articles alone with just network in the title, right? Network throughout the article is probably going to increase that number by another half or so. And this is published in roughly the last 20 years. The players in this field are CS, math, and stats, certainly. Some of the other quantitative fields like signal processing, stat physics, but then you start going beyond to the domain areas, even going down to areas like digital humanities or something like that. So there's really, networks are really pervasive everywhere in the fields. Right? So if you say, where's statistics in this? Right, the answer is everywhere. Right? Really, statistics is in any of the data-oriented aspects of network type analysis. As a statistician trained, one of the things I tend to do is try and apply the template or perspective that I was trained with in order to talk about problems in this area and the challenges we face. So I think about sampling and design as paradigms. I think about description and visualization. I think about modeling and inference, prediction, and whatnot. And a lot of the problems in this area actually have sort of two flavors to them. You can talk about network analysis of a network, where you're literally thinking about, a network like in a fishnet, what is it about this net that I want to know something about? Or you might be talking about network analysis on networks. Say you're talking about a communication infrastructure and you're doing anomaly detection on the communication passing over that infrastructure. Right? So there's sort of nuances of both here. I will probably talk mostly about a handful of problems regarding of networks and not so much on. All right. Broadly speaking, the challenges in this area, right, come from three key factors. Right? And this is why the subtitle is not just visiting the foundations and kind of nice reminiscence, but really revisiting, because we're still working on them. Right? One is the key fundamental aspect of network data, that it's relational. Right? So you think about IID data independent, right? You think about temporally correlated data, you think about spatially correlated data, and this is something different yet again, right? This is data where fundamentally you're talking about relations. It might literally be a social relation, or it might be some structural type of relation, like the fact that two roads intersect, or you have different stations on a metro system or something like that that intersect, right? The second thing is, going with that, there's typically a lack of geometry. Right, I'm going to talk about geometry a little bit, so I'm going to at least quibble here and say lack of traditional geometry. If you think about a network in the sense of a bunch of things that are connected by links, and you think about a proverbial fishing net or something like that, if I take that fishing net and I throw it on the ground and I go pick it up later, right, it's almost certainly going to be hanging in a different way depending on where I pick it up. It's the same fishing net. Right? So the idea that if I have a face or an object or something like that, and they're 3D or 2D, they're the same thing. And they typically, up to some small set of transformations, right, are basically the same thing. They exist in a geometry. We're used to thinking about them in geometry. And that geometry then goes to issues of scalars and vectors and matrices, that all is the underpinnings of a great deal of the type of analysis that we do, right? Linear algebra and all that. Once you take that away, you have to figure out a way to get back into those contexts for certain problems where you want to do familiar analyses. But the starting object is this net hanging in your hand. Right? And then finally, of course, there's a big data aspect to a great deal of the problems that are being looked at from a network perspective, because again, we're often talking about a systems level context, so you're talking about systems level data. All right. The reason I'm talking about foundations is because despite a fairly well-developed ability to talk about things like IID data, spatial data, temporal data, right, there is a new need to kind of revisit a lot of the things we're familiar with for the particular animal that is networks. And we've been doing this for 20 years. But the part of my talk here is going to be, there's a number of things that were left behind that are really quite fundamental. They've been left behind partially, I think, because statistics didn't engage nearly as much as certain other fields, and also because they're hard. Right. So where are we? I'd say we're somewhat on the way to where we want to be, but there's still a long way to go. All right, so I'm going to try and convince you of that, and at the same time, as I say, give you a little sense where we are by some vignettes. Yes, please.
So we understand the qualifier, you're referring here to complex networks. Do you mean large, a certain degree of connectivity, non-planar? Could you define what
Sure.
that's supposed to include?
Sure. So I gave you 3 examples earlier. Any of them could be that, right? So you could talk about a gene regulatory network. You could talk about some sort of collaboration or co-citation type network. The router level network is it's a logical topology, but still it's a logical level type network. I wouldn't necessarily be referring to something that's restricted to be planar or something like that. Generally, in complex networks, they mean a graph. Right? And as to whether I actually require characteristics of the network to be obtaining here, we'll talk about sparseness a little bit in this one here. Here we'll talk a bit about heavy tails. And here this is a different animal. I'll show you what we're talking about there. But none of what I'm talking about here is particularly as focused as I think you're thinking about. Yeah. All right, so 3 vignettes here. I'm going to try and give you a sense of what I mean by foundations, why there's a novelty. All the problems I'm going to talk about are very classical problems. They're things that we would want to do every day with regular data, and in fact we want to do every day with network data, only our ability to do it is currently evolving. Right? And in doing so, hopefully you get a little sense as to how and why of the challenges in this area. All right, so 3 topics: adjusting for bias in network sampling. If you have a network and you only see part of it and it came through some kind of sampling process, for interesting reasons there's going to be non-trivial bias in that network. How do we adjust that away to get a more unbiased picture of what the network actually looks like? Right? Propagation of uncertainty. Right? Those 500,000 papers that are out there, probably 2/3 of them alone are applied papers. Right? Not meaning that in a derogatory sense at all, meaning that it was good, solid applied analysis where the central part of it was to produce a network, report on the network, and summarize the network. Right? But a large fraction of those have noise in the underlying fundamental measurements with no sense of propagation as to the uncertainty to the final statistics that are actually reported. Right? So propagation of that noise is a non-trivial problem. Finally, in the area of complex networks, for most of the 20 years, it's been very common to focus on one network, typically one large one. But we have been moving in the last 5 years towards an era where we have many networks, sometimes thousands of networks. So we're moving to a point where individual data points are actually networks entirely. And you get now led to a question of what is the large sample treatment of networks? Can you talk about an average of networks? Does it even make sense? If you have an average, can you do things like tests or confidence intervals? Does that even make sense? So I'll give you a little sense as to what that is. All right, each of these are going to be fairly quick-paced. They're mainly just meant at telling a story. There's papers behind all of them. I'm happy to talk more about all of them, but I figured the most efficient and useful thing I could do for your folks was to sort of show you a number of different snapshots. Okay. All right, so network sampling. Right. The idea is that you have a graph G. G consists of a set of vertices, so the vertices might be people, right? And it has a set of edges E. The edges might be some sort of declared relationship. Think Facebook, if you like. Right? Or if you don't like, there's other platforms we could talk about. Right? Somehow or another, you have only a subgraph. There are a lot of ways you could talk about doing sampling. I'm going to just picture the idea that it's literally a subgraph. If you see a vertex, it is a true vertex. If you see an edge, it is a true edge. And conversely for a non-edge. We'll talk about noisy observations in the next vignette. Right? So through this sampling, you want to try and get some understanding of the characteristics of the graph G, but you only have a subgraph. Right? So you could picture very extreme ways of getting subgraphs that would give you very extreme, unrepresentative senses of what the actual graph looks like. Right? So the sampling matters. The sampling also tends, even when it's fairly simple examples, to induce non-trivial behavior, right? So here's an example. If I go through and I flip a coin for each of you, and I get only a handful of you who come up at heads, and I say, you're going to be the vertices in my subgraph. And then I'm going to ask, do you have a link on Facebook, right? That's called induced subgraph sampling, right? The simple random mechanism is actually choosing individuals, but it induces a sampling of edges. There's other analogies in communication where they do so-called trace route sampling, which are path-based sampling. Those get even more non-trivial in the type of biases where you see certain parts of the network instead of other parts of the network. Right? The attempt to try and take classical sampling and use tools from classical sampling to figure out how to adjust for the biases that I just told you about goes all the way back to the 70s or 80s with Ove Frank. Right? But what he did there was really relevant only to a handful of things, like the number of triangles, the number of edges, fairly simple descriptive statistics. Right? Other characteristics are much more common in the modern era, and some of them have been found to have extreme forms of bias. So the degree distribution is one of them. Degree distribution, degree of a vertex is how many links it has. How many friends you have in Facebook is your Facebook degree. Facebook's degree distribution is literally the statistical distribution of degrees across all people. Okay? So it turns out in 2000s, right, with a series of papers, right, they found that sampling of networks can induce extreme biases to the extent that when often people are looking at internet-type networks and seeing very heavy tails to degree distribution, saying that there was some extreme connectivity, right? You could actually induce that in a graph whose true distribution was very tight, purely by sampling. Right? So that's quite an artifact. Right? So we worked on a problem where we said under a certain collection of sampling designs, it's certainly by no means all of them, you have a very simple relation between the distribution of degrees as a vector I'll call n and the expectation of the degrees that I actually see. So you have a true network, you have a subgraph. The subgraph has a degree distribution, the true network has an actual degree distribution. The relation between them is through a linear relation with a matrix P. All the sampling is embroiled into that matrix P. Right? We're not going to go into details, but depending on your design, P can be a very stable matrix to invert. It can be a very unstable matrix to invert. And that should already give you sort of an intuitive sense as to how biases can be in this kind. Franck proposed in '78 the natural estimator here, which says, well, invert P, right? And keep your fingers crossed, right? So the problem is here that P is typically not invertible in practice for the designs we talk about, and non-negativity of the solution isn't necessarily guaranteed. Right. This is a simple example. This is an example of a very tight distribution of degrees. It's for a network of 100 vertices, 500 edges. It's drawn at random from the urn of all networks that have 500 edges on 100 vertices. And this thing is highly well studied mathematically and known that it has almost a Gaussian-like distribution. Right? When I try and sample it with 60% sampling of vertices, which is very, very generous, and then I invert the matrix P that I had, I get something like this. This is negative, if you can't see, right? Degree distributions don't go negative, they're distributions, right? So what you're getting is intense oscillations here due to the instability of the matrix inversion, right? So not a good idea. Even publishing in '78, Frank said it's not a good idea, right? But he published it anyways. What we've done here, this is my one slide summary of obviously a much larger paper, right? What we did is we said, if that's an unstable inversion in the modern era, what you want to do is regularize that inversion, right? So we set up a regularized, weighted least squares problem, where the n star minus Pn up there is taking the idea that in expectation, the observed degree distribution is some function of the true one. So it's exactly using the formula Franck used, right? Cast in a least squares framework, sticks on a penalty to induce smoothness or whatever other characteristics you want. Plus says the degrees that we get should actually be greater or equal to 0. Right? It turns out there's relations of a lot of quantities in graphs. A very simple one is that the total number of degrees obviously have to equal the number of vertices in the graph. Right? So you turn it into a constrained optimization, which is something that in our modern era we're very good at, and is very much a go-to in statistical machine learning for doing estimation problems. Right? When you do that, you do the programming behind it and you implement it. Here's an example of how this works on relatively large networks. These are all social media type networks. Some of them are platforms that had their raison d'etre more about 10 years ago than now. I have a 16-year-old. I can't even keep up with all the different changes in platforms, right? But here we have Friendster, Orkut, and LiveJournal. These are degree distributions on a log-log scale because these are all real social media platforms with the canonical heavy tail to them, right? So we have a log-log scale. And then we have here in blue on each of these plots is the true degree distribution for the network that we have, right? So we're doing a synthetic study here where we take observed data, we do our own sampling, and then we try and estimate again because we know the truth, right? In black is what you get when you do the sampling. Here I think we're doing sampling at 30%. We have results in the paper going down to 10% and still getting essentially the same type of results here, right? Red is the estimate of the true degree distribution, right? And so if you back off and say what's going on, basically you have blue. On a log scale, you have a sizable shift to the left in black. Right? And on a log scale, that means you are way off, right? And then in red, we put you right back on top of the blue for a large number of these cases. Okay? So Moral of the story, it's possible to invert these biases if you have enough understanding of the sampling mechanism that was involved and how it relates what you're after in the real graph with the graph that you actually see. But you have to do some work. It's what they call unequal probability sampling, so it puts you in the second half of a traditional sampling textbook right off the bat, even for the simplest sampling design in networks. If you want to have a higher level statement as to what does this get you, this is a lot of text and images, but basically it's saying that once you have a degree distribution, you can talk about degree moments, and degree moments end up bounding all kinds of interesting quantities, including epidemic thresholds. So if you have a friendship network and you're wondering about the flow of rumor in a network, and you'd like to bound using some basic epidemic spread models, the epidemic threshold, it turns out you can bound that epidemic threshold based on moments of the degree distribution. This is in 3 of the different networks that we looked at. The actual epidemic threshold in red, right? 2 different upper bounds and a lower bound coming from inverting these bounds here. This is our ability to estimate those bounds. So we're able to really nail those bounds. The gap with the epidemic threshold is mathematics, right? That's an inherent gap in the technique there. Okay. All right, any questions about that vignette? That's vignette number 1. The other 2 are going to kind of go similarly. No? Okay, so we'll move on. So vignette number 2. In the sampling we just talked about, we said if you see an edge, it's an edge. If you see a vertex, it's a vertex. Now let's change that up. Right? So the idea is that very often what we see in network analysis is you have some system and elements and interactions you're interested in. Right? This is you're thinking at a systems level. Right? I'm working on the internet. I'm working on traffic. I'm working on whatever have you. Right? You say, let's collect some sort of notion of measurements that represent the elements and their relations. So collect information on people, collect information on routers, right? Collect information on traffic. Right? This is at 10,000 feet sort of the recipe that most network analyses are doing. Eventually you get to representing that collected data via network. That might be an extremely intensive task. It might be ad hoc that you decide when there's edges. You might do some fairly formal statistical inference. At the end of the day, you have, as a function of the original data, a network. And that's typically now what people say, now here's my network. Right? And they go and they start summarizing the properties of that. They say, this is how many edges, this is how many triangles, this is how long the shortest paths are, this is if it's connected or not. And they start associating those structural characteristics with questions they want to know. If it's not all connected, it's in pieces. What are the implications, say, on the flow of traffic in the network or something like that, right? So that's kind of at 10,000 feet what people are doing with network structure is they're matching structure with questions and characteristics, right? And this sounds good, right? Except here's the obvious, right? If there's uncertainty in determining the edge status because of uncertainty in your original data, right, where if you took measurements again, you would get slightly different measurements and therefore perhaps slightly different determination of edge or non-edge, right? Then there's uncertainty that's going to propagate to any statistic of the network that you cite. You tell me what the average path length is, and then on another day with slightly different data, you're going to get a slightly different average path length, right? So why in the literature do we not ever have something that has number plus or minus standard error? There's been surprising little work on this. We could talk about different reasons. Some contexts have very little uncertainty. Right? Other contexts have a lot. Right? The other thing, though, is that it's a very hard problem, it turns out. Right? So work is needed on characterizing this propagation of error and then adjusting for the error to actually try and create estimators. Right? And this is really, I've said for a few years now, we're beginning to make progress, and I'll show you some of that, but this is really very much a missing foundation in network science. Here's an illustration. So if you work in computational biology, right, and you've worked with gene expression data, you've seen these types of heat maps. Right? So the heat map here is showing for a whole series of different experiments what the relative activity, let's call it, it's gene expression levels, typically on a log scale, of genes were across a whole bunch of genes. There's about 400 genes here. I think there's about 40 or so experiments here. And typically they're looking to see if there's correlation, right, gene by gene across experiments. So you see kind of a patch here where this patch of genes is doing something similar here, something similar here, something similar there, right? If they're doing similar behavior, then something about the dynamics of them is similar. They're probably participating in common function in whatever cell they're in. If they're doing that, that's highly suggestive of regulatory relationships for various biological reasons, right? So people have a whole sub-area, there's a whole sub-literature just on constructing networks from this type of data, exploiting these correlations that we see. This is one very simple one, just based on notions of literally using Pearson correlation. I get a bunch of different genes with a bunch of different edges and a bunch of ones that don't have edges because I infer that there were or were not, right? How I did it doesn't really matter for this talk. If you didn't like this one, I have 100 others I could show you. That's how large and active that literature is. The point is that if they do these experiments again, I will get a slightly different network. So if I want to now talk about the density of edges, how many edges do I have compared to how many I could, which is n choose 2, right? Here it's 0.14. 0.14 plus or minus what? We have no plus or minus. Okay? So here's a general formulation, right? We have some network G. Right? Say it's undirected for the moment. We have eta as some kind of summary. Hold on to density if you like. The density of edges in the network. It could be the density of triangles, could be shortest path lengths, any favorite number of yours, that's what eta is. Right? We've been working typically with counting subgraphs because it's kind of like an average, and average is the go-to place for a statistician. We know how to work with averages. With a little luck, we can make progress in new problem areas with averages. So instead of having G, we have this noisy version. We're going to say we know the vertices, make the problem a little easier for ourselves, and it's just the edges that are not known necessarily. Right? The natural way to model this is with a bit flipping type model, right? Coin flips. Right? So Yij says whether or not there is or isn't an edge between vertices I and j. If there truly is one, right, then I will say there is one Bernoulli with probability 1 minus beta. So beta is my type 2 error in statistical parlance. Right? If there is not an edge in reality, right, I'm going to say with probability alpha I see one anyways. That's your so-called type 1 error. Right? So the exact analog of type 1, type 2 error is present in these network contexts. And if you simplify things enough, you can kind of write down a simple model. Our goals have been to understand how the propagation of these errors from the true edge, non-edge sets to what we actually see, how that propagates to some kind of summaries, like the average density in the network. Right? And then second of all, improve upon the estimates. So we've made some progress. One of the things we can do is we can characterize the distribution of the discrepancy in the case of large graphs and lower error rates. Right? In a nutshell, again, I'm not going to show theorems and proofs and all, right? In a nutshell, the results show that the discrepancies exhibit Poisson-like behavior. That's coming from low error rates and large graphs, similar to the way you think about Poisson coming from binomial coin flipping when you have many coin flips of very low probability of showing up, right? Only you get what's called a Skellum distribution because you can get two types of error, so you actually can be off in one way or the other way in terms of accuracy of estimation. In other work, what we've done is two things. First of all, we've proven the impossibility, which if you think about it is not hard, but if you don't know the type 1 and type 2 error rates, and you don't know the number that you want to estimate, say the density of the network, you have three numbers, one graph. Right? Now you could say, what's your sample size? I have a paper actually written that literally is what's the sample size when you have a network, right? Because we actually had a dinner conversation after drinks, et cetera, and we had three opinions on what happens when you have a network. Right? One was that your network is an adjacency matrix. There's n choose 2 entries in that adjacency matrix that are unique. It's roughly n squared, so your sample size is n squared. Another person said, no, you have n individuals. So your sample size is n. That's an order of magnitude different. Then we had the final answer. Someone said, it's 1. Right? Because you have one network. So it's actually not trivial to ask yourself, is it impossible to estimate 3 numbers from one perhaps very large network? Right? But because of the nature of those numbers that we're after, there's an impossibility theorem that says, given unknown type 1, type 2 errors and these subgraph counts, you cannot estimate all of them. On the other hand, we've also developed a method of moments estimator for the case where you do have sufficient number of replicates, because the typical answer to those impossibility theorems is to have replicates. And so it turns out that under appropriate conditions, with as little as only 3 replicates, what you need is you need 1 for each of these numbers. Right? Then it's possible to estimate the type 1, type 2 error rates, right, as well as low-order subgraph densities in a manner that's statistically consistent, meaning converging with probability 1 with large networks, and asymptotically normal. The asymptotically normal is really key, right, because that's where you're going to get your standard way of doing plus or minus 2 standard errors and get your confidence intervals at last, or do your tests or whatnot. Right? So here's an example. We took the log gene expression data similar to what I showed you before. This is an E. coli. We have 153 genes, 40 different experimental conditions. So the sample size from this point of view is 40, working with vectors of length 153. But it turns out, and this is very common in biology, they did 3 replicates. We had actually, with the students and I had a talk on the car as to why do we have 3 replicates, right? Well, because if you only do 2 and you want a mean and a standard deviation, it's just a 1-to-1 mapping. But resource-wise, they don't want to do much more, so they do 3. Right? So with 3, it turns out that's enough. Okay? For each set of replicates, we constructed the same correlation network that I was talking about earlier. We used a nominal 0.05 Bonferroni corrected error rate on the edges. Right? This means that edge by edge, it's a much smaller error, but the probability of making a mistake of a certain type on the network as a whole is only 0.05. The empirical edge densities we got from each replicate were 0.073, 0.075, 0.074. So roughly 0.07 consistently through the data if you just took the empirical number that you get from the network. If you instead try and estimate type 1, type 2 error, and then estimate density corrected for it, you get, first of all, your type 1 error is about 2.5%. Type 2 error is about 25%. Right? And finally, your estimate of the network density is 6.7 instead of about 7.4. Right? With a 95% confidence interval of about 0.062 to 0.072. So you see that this thing is coming in just shy of the numbers you have there, giving you fairly strong evidence that these are actually overestimates when you just quote the density of the network that you get instead of trying to actually go through a process of correcting for error. All right, any questions on that vignette? Now, these are kind of fast and furious, but am I leaving you with a bit of a picture as to kind of what? Okay, good. That is always my goal, particularly with mixed audiences, with different backgrounds and whatnot, is just to live a picture, and people can reach out, they can ask questions, you can dig in wherever it is you see. All right, one more vignette then. We're going to change gears, as I alluded to earlier, from one big large network to many networks. Right? This is really in a number of areas becoming where we're at. It almost seemed like heresy in complex networks at some point 5 years ago when it was first coming on. It's like, more than one network? Well, that's easy, because that's just like what we know how to do, IID sampling. So we know how to do it. That's not the hard problem. The hard problem is one big network. Turns out this one's a touch more harder than people thought. Okay, so increasingly in computational biology, we're getting networks for every single individual of their expression relations. We're getting, in computational neuroscience, networks of the way the brain is communicating in every single person. Right? And there's a variety of other contexts. These are sort of IID type ways of having many networks. There are all kinds of contexts where you have temporal time series of networks. And you can think of that as a temporally correlated way of having many, many networks. Right? So really facing a need now to be able to ask questions like, what's the average of a collection of networks? And I put it in quotes because maybe that's not even the right word. But we all kind of know what we mean when we say that. I got a lot of networks. What are they like on average? Right? Once you start going that direction, there's a whole toolkit from Stat 101 that you could start using to list a bunch of questions, right? Do networks differ on average from a certain nominal network? That's a one sample test, right? Do two collections of networks differ in cases and controls? That's a two sample test, right? You see where I'm going, right? What's next? Regression, right? So you could say, what factors are going to contribute to difference in networks? Can I do a regression of a network on various factors of interest? Right. This falls into the larger realm where a lot of data science is going these days in terms of structured data objects, right? Where an entire video is considered a data point, right? Or entire image, if you want to simplify, is considered a data point, right? Entire genome is considered a data point because we have them now for thousands of people, right? All it does is it raises the sea level on the notion of a data point, and it moves to sort of a meta level and says, I actually don't want to worry about the details of which gene connects to which gene. I just want to take your whole network and treat that as an object. Okay, so this is one instantiation of that much larger change in data science in the context of networks. Okay, so in a nutshell, are you getting a theme here, right? Because I told you I'm talking about the cutting edge stuff. So we lack, again, certain things here. We lack principal analogs of even the most basic 101 tools. Right? And this is what my group and a number of others have been part of what we've been working on the last few years. Right? So here's some questions you could ask. It's much harder to answer those questions. Right? So we have some answer. What it involves is typically trying to deal with the fact that we don't have Euclidean objects. So if you want to start with the simplest question we had, what's the average of a network? And from there, try and build. You have to say, if I have a bunch of fishing nets lying on the floor, right, and I pick them up, how do I average fishing nets? It's not clear, posed that way at least, right? So we have a set of results that does the following. Right? First of all, we throw in some geometry. We show that certain classes of networks can be associated with so-called nice subsets of Euclidean space, so formally smooth manifolds. We then throw in some shape analysis. Shape analysis has to do with some of the type of things that are done in vision. Also has to do with doing analysis, say, on a sphere, where you really want to stay in a sphere instead of thinking of the sphere as being in 3D space. Right? So in there, we exploit what we learned in the geometry, right, to design a corresponding natural distance. The natural distance is used to employ a non-parametric notion of mean. Right? The non-parametric notion of mean simply says, you give me a set of objects, the average is the one that is closest to all of them, based on some notion of distance. Okay? So the distance and the geometry go together. Given that you have a distance, the distance and notion of mean go together. Okay? Once you have a notion of mean, you can start working on the probability theory. Right? So we have an associated asymptotic theory, meaning formally we have a CLT. So we have a central limit theorem telling me how this average behaves for large samples. Central limit theorem is sort of the foundation of all the Stat 101 tools we get. Your z-test, your t-test, your confidence intervals, your chi-squares, they're all based essentially on a central limit theorem. Once you have that thing, you can crank out a number of the Stat 101 things. And so finally, here we are at statistics, right? We have analogs of classical 1 and 2 sample hypothesis testing. Formally, it's in the spirit of Hotelling's t-squared test. Okay. All right, let me give you an illustration of that, and then I'll wrap up. Okay. So one place you can get a whole bunch of networks, and they're online and available and whatnot, is the so-called 1,000 functional Connectomes project. So the Connectome, every subfield now wants to name networks of their own type in a certain special way, right? So within neuroscience, they call the Connectome the functional brain connectivity networks that they've been using. These are always inferred from some sort of measurement type data, typically imaging data, so fMRI, MEG. You might have local field potential type data in terms of EEG or ECOG or something like that. So there's a variety of measuring modalities. They've done this on, as you can guess, 1,000 people. Right? And for those 1,000 people, you can take those raw measurements and create networks. Now, just like the last vignette, those also have uncertainty. Here, I'm going to treat them differently. They have uncertainty. I'm just going to treat every network as a draw from some distribution. I don't even need to know what the distribution is, other than it has, it's not totally bizarre, because I'm going to do a central limit theorem. Right? So it's just like we're used to in standard type analysis. Right? This is a way of representing the connectivity in a graph. This is an average. To simplify, it's a visual representation of the average for the females, right, which were slightly more than the males in the 1,000 functional connectomes group. And this is a visual representation of the connectivity in the males. Right? And what this does is it takes regions of interest in the brain. They parcellate up the brain. They assign a number to them. We line up the numbers along the rows and the columns. And then it's black if there was an edge between them. The edge means some kind of dynamic connectivity. Typically, like there's some sort of signal, and at least at some lag or another, there's another signal in the same brain region. Right? So it's a temporal version of the correlation we were talking about for the gene networks. Right? So this is showing the adjacency matrix for females on average, males on average, and if you difference them, that's a minus sign, then you would get this. And you'd say, okay, is this telling me then that this is the differences, right? Well, based on about 600 and 400 people, that's giving you some reasonable evidence that there's some differences there in connectivity. And if someone were really doing this analysis, you would then start obviously taking into account what are the actual regions of the brain involved. Those regions form bigger regions together, so you'd be looking at what's going on in the different clusters and associating that with the neuroscience knowledge that you have. All that, obviously, I'm just sweeping under the rug here. Right? The value added in what we've done here, right, is unlike what they call mass univariate techniques, where they want to test, is this a statistically significant difference? Right? If I treat the 1,000 as a sample from the overall population, then either our technique or the simpler techniques that are out there, they all say, yes, this is statistically significant. But if you push us and you say, I don't really run 1,000 sample lab. I can get 10 people, max, maybe 20, right? which is the typical story for most labs across the country, right? Then the standard techniques are unable to tell that this is actually what's going on, whereas ours still can. And the secret there is that the standard techniques, what they do is for every single cell, they do a test. It's a simple test of difference of means. And then they try and adjust for the fact that they're doing thousands of tests. So they do a Bonferroni correction or something like that, which means that you apply a really extreme threshold for significance. And by being so extreme and conservative, this all goes away. And you say, there's nothing that's such a strong signal that I can confidently tell you it's there. Our methodology is essentially multivariate statistics. So it tests the entire thing at once, exploiting the correlations in network structure across subjects. And in doing so, you spend your type 1 error much, much more wisely, to the extent that you can push those sample sizes down to around 10 people per group. All right. This is an area where we have a great deal still going on. We've been working in personalized medicine and multi-omics. We're now actually, instead of one network, we have layers of networks, so-called multi-networks. Multi-networks where you have a similar vertex set, but you have different notions of connectivity. So if you have people, we might say one layer is if you're connected on Facebook. Another layer is if you're connected on LinkedIn. Another layer is if you're connected on Instagram. Okay. A lot of you may be connected, or you may not be, but if you're connected on one platform, you may be connected on the other platform, you may not be. That's interesting. And that's a correlation structure. That's a notion of a multivariate network. Right? So we have an extension to that with particular applications of personalized medicine where they have multi-omics measurements on people and measure their gene expression, measure their protein activity, measure all that. Everyone is a layer in the networks. At the same time, we've been looking at the notion of egocentric social networks and privacy. So this is the context where now you have networks, but you don't have any labels on the nodes. So if I want to average things, and I have, say, a network of your 10 closest friends that we then privatized, right? So that I have a network of 10 nodes for you, and 10 nodes for you, and 10 nodes for you. I don't have any labels on that. How do you align those to do anything like an average? Right? So really you're working with some sort of version of the previous problem that passed through the permutation group of all different ways that you could actually align the networks. And formally, that is actually sort of what's going on. This has tight connections to privacy, which is one of the main reasons why you would actually have those node labels go away. Right? So formally, it corresponds to collections of unlabeled networks. And again, we have work on this. All of those extensions have to go back to the beginning and say, let's set up the geometry so we can set up the mean, so we can set up the probability, so we can set up a central limit theorem. The geometry gets much harder, for example, in the case of unlabeled networks. This, I'm not expecting anyone to sort of grep what's going on there, except that that's a visual representation for just 3-node networks when you're removing the labels. Right? You're seeing that it's a very particular sort of structure with particular relationships. It turns out edges matter. Vertices matter. They're all subsets of different types of networks, and you're trying to do probability on an object like that, literally on it. Okay, some closing thoughts here. Using this network-based perspective in modeling analysis really is pervasive now. Right? Much of this work done in the area is very problem-specific. Right? So this is nothing in my talk is meant to somehow diss any of the work that's been done in 20 years. There's been amazing work that's been done in the different fields. But as a statistician, I can both look at that work and appreciate it, and I can also go through and look and go, you know, there's a number of foundational things that we've left behind, and I can gather those together, structure them, and start laying out a program saying, these are some things that we need to do. Right? Particularly when I talk to statistics type fields, I hope there's actually a hook there, because I put that out and say, isn't this interesting? Right? And sometimes they go, yeah, and sometimes they go, oh, yeah, I don't even want to tackle it, right? But not only are these problems broadly relevant, right? But as you could probably see from what we're using here, we're often using a lot of cutting edge techniques and technologies that are already at the interfaces of a number of other fields. So bringing in non-trivial geometry. Geometry is much more present in the cutting edge of the analysis of structured data than it has been for years, because we've been exploiting Euclidean, right? We've been exploiting Euclid's results up till about the last 10, 20 years or so, right? The idea of bringing the constrained optimization, which is very much a development of statistical machine learning era, to bear on a problem that's previous only real major improvement was 1978. Right? So this is what I mean by a lot of these problems we're working with are in very fascinating ways touching on the exact tool sets that are being used and founding to be the right thing now on a number of different areas. Okay, I'm going to thank all the people. Of course, this is my sort of overview way of trying to thank the various people who've actually allowed me to get this work done, and of course the people who supported. So, and I think I'll stop there. So.
Thank you. Does anyone have any questions online or in the room? There's some clapping.
Thank you for that.
If no one has any questions, I think we will break for lunch. Thank you.
Thank you.